Dear students, Assalamu alaikum. Today we are going to talk about our course Physiology and Immunology, course code CHEM444. This is lecture number 16 of the series of our online lectures. Today we are going to talk about types of antigen antibody reactions. In the previous lecture we talked about antibody antigen reaction, that it was kind of an introductory type of lecture. In this lecture we talk about details of the different types of antigen antibody reactions. The outline for today's lecture is we will talk about precipitation reactions, we will talk about agglutination reaction, we will talk about complement fixation, we will talk about enzyme linked immunosorbent assay ELISA, we will talk about immunofluorescence. So these are the different types of antigen antibody reactions. So this was the outline. Now we move towards our lecture. Precipitation reaction. When a soluble antigen combines with its antibody in the presence of an electrolyte NaCl at a particular temperature and pH, it forms an insoluble precipitate of antigen antibody complex. The antibody causing precipitation is called precipitin, and the reaction is called as precipitation reaction. So here you can see the antibodies. Here are the antigens, and here is the antigen antibody complex. This antigen, you can see the red, the antigen, and the blue color is designating the antibodies, and the Y-shaped structure you can see here. This is here like this, and here it is like this. So this is antigen antibody complex. Function of precipitation reaction. Precipitation occurs in two media, liquid, gel. Precipitation in liquid. Antigen antibody reaction performed by placing a constant amount of antibody in a series of tubes and adding increased amount of antigen. Antigen antibody reacts together, resulting in precipitation. Plotting the amount of precipitate against increasing antigen concentration yields a precipitation curve. Here is the antibody access zone, here is the equivalence zone, and here is the antigen access zone. So supernatants, excess antibody, excess antigen, antibody precipitated, and antigen added. So this is how this plot looks like. Precipitation curve shows three zones: zone of antibody access, antibody access zone, you can see in the yellow, zone of equivalence, this is the zone of equivalence zone, and zone of antigen access. You can see here is the zone of antigen access. So precipitation in gel, radial immunodiffusion, Mancini. In these methods, agar gel or similar gels are used on plates or petri plates. Both antigen and antibody diffuse freely in the gel system in all reactions at a certain point, depending on the rate of diffusion and concentration of the reactants. A zone of equivalence will be formed, which is seen as a visible precipitation. If antigen or antibody preparations are complex, multiple bands form. These are again of two single types. These are again of two types: single diffusion methods and double diffusion methods. This is radial immunodiffusion, antigen diffusion. Antigen is shown here. Precipitate forms ring. Antibody incorporated in agar. Double immunodiffusion, antibody, antigen. Here is the precipitate. Here is the agar matrix. These are precipitation reactions in gels. Agglutination reaction, when a particular antigen is mixed with its antibodies in the presence of electrolytes at a suitable temperature and pH, the particles are clumped or agglutinated. The antibodies of the serum causes the cellular antigens to form clumps, and these are called agglutinins. The particulate antigens that are aggregated are termed agglutinogens. Slide agglutination. This is a rapid method to determine the presence of agglutinating antibodies. 1 drop of positive control reagent is added to circle. 1 drop latex reagent is added to each of 3 circles is shown. Disposable test slide. So 1 drop of negative control reagent is added to circle 2. This is the negative. The calories are quickly and correctly emulsified into reagent in circle number 3, and the slide is rocked by hand for 45 seconds and placed on a slide rotator for another 45 seconds. This is showing slide agglutination. To uniform suspension of particulate antigen, a drop of saline is added and then a drop of antiserum is added. The slide is gently rocked or a fine loop is used to mix the contents. If granulation occurs, the test is positive. If it takes a minute for the test to complete and is visible to the naked eye, sometimes confirmation may be done by observing slide under microscope. This test is used for blood grouping, hemagglutination, and cross-matching. Hemagglutination inhibition. Patient sample, human IgG expressing the G1 epitope plus anti-HU IgG G1 epitope specific. No agglutination. All available anti-G1 epitope is bound by free G1 expressing antibody in the patient's serum sample. This is a positive reaction. Human IgG with other heavy chain isotope, patient sample plus G1 epitope specific plus RBC agglutination. This is a negative reaction. Tube agglutination. This is a standard method for quantitative estimation of antibody. The serum containing antibody is diluted serially with saline in several small test tubes to which a constant volume of antigen suspension is added. A control tube is kept which has no antiserum. The tubes are incubated until visible agglutination is observed. The tube showing highest agglutination is referred to as the titer. Tube agglutination is employed for the serological diagnosis of typhoid, brucellosis, and typhus fever. Pedal test is used for the estimation of typhoid fever. In this test, antibody contents of the patient's serum is measured by adding a constant amount of antigen Salmonella typhi to the serially diluted serum 1/20th, 1/40th, 1/80th, 1/160th, 1/320th, 1/640, and this is the control. This is titer. This is 160. This is the example of tube agglutination. Passive agglutination test. It is similar to hemagglutination test, but the physical nature of the reaction is altered. The antigen is coated on the surface of a carrier particle and thereby helps to convert a precipitation reaction into an agglutination reaction, making the reaction more sensitive. The carrier particles used can be RBC, latex particles, or bentonite. Sometimes RBC coated with polystyrene tanned RBC can be used. When patient's serum is mixed with these, it leads to agglutination. This test is used for the diagnosis of rheumatoid arthritis. Carrier particle, soluble antigen, coated particle. So coated particle, antibody, and agglutination. This is passive agglutination. Agglutination inhibition provides a highly sensitive assay for small quantities of an antigen. Example, first home pregnancy test, kit reagents HCG and anti-HCG antibody, heptane carrier conjugate, test procedure, urine plus anti-HCG, incubate plus HCG carrier conjugate, observe for visible clumping. Possible reaction, reaction not negative, reaction not pregnant, positive reaction pregnant. So here is this carrier conjugate plus antibody, visible clumping, HCG in urine plus antibody, no visible clumping. So reaction, negative reaction, this negative result, and positive reaction is showing the pregnancy. Complement fixation, lysis of RBC or bacteria requires some non-specific unstable components of fresh serum which are called complement. This complement system comprises of 11 proteins and are present in every individual. They bind to Fc component of antibody involved in antigen antibody complex. This ability of the antigen antibody complex to fix complement is used in complement fixation tests. In the first stage, the test antigen and the antiserum heated to 56 degrees C to inactivate complement are mixed in the presence of known amount of complement. This is incubated at 4 degrees C for 18 hours. If antibody specific for the antigen is present in the serum, antigen antibody complex will be formed that will fix the complement. Complement fixation test, serum with antibodies, serum without antibodies. Antigen binds with antibodies, unbound antigen. Complement binds with antigen antibody complexes, unbound complement. Homolysis in sensitized red blood cells serve as an indicator. Hemolysis in sensitized RBCs serve as no indicator. RBCs clot into a pellet, no lysis. RBCs lysed by unbound complement lysis, non-reactive. So this shows the reactive and this shows the non-reactive. ELISA, enzyme-led immunosorbent assay. In 1971, enzyme labeled antigens and antibodies were developed as serological reagents for the assay of antibodies and antigens. These are very simple, sensitive, economic, and less hazard when compared to RIA. The ligand used here is a molecule which can detect the antibody and is covalently coupled to an enzyme such as peroxidase, beta-galactosidase, alkaline phosphatase, etc. ELISA is of three types. Indirect ELISA. This technique is used for the detection of HIV. The envelope proteins are developed by recombinant technology and coated on the surface of the microtire plates. Suspects serum is added and unbound proteins are washed off. Sandwich ELISA, used to detect the presence of antigen in a sample. The well is coated with antibody specific to the antigen and then suspect serum is added, allowed to react. The wells are washed to remove unbound antigens. Here is the example of how the indirect ELISA function. Here is example of how sandwich ELISA function, and here is the example of how competitive ELISA function. These are the different types of ELISA. So you can have a look at the how it functions. Antigen coated well, then wash, add specific antibody to be measured, add enzyme conjugated secondary antibody, and then wash, add substrate and measure color. So this is the example of indirect ELISA. Sandwich ELISA, antibody coated well, wash, add antigen to be measured, wash, add enzyme conjugated secondary antibody, wash, add substrate and measure color. This is the example of sandwich ELISA. Incubate antibody with antigen to be measured, add antigen antibody mixture to antigen coated well, wash, add enzyme conjugated secondary antibody, wash, add substrate and measure color. Then a labeled antibody against a different epitope of the antigen is added. Unbound antibodies are removed by washing, and this is followed by addition of colored substrate and development of color. The intensity of color is directly proportional to the concentration of antigen in the serum. Competitive ELISA, like I explained here, so you can have a look at. Now we go to the final type, the immunofluorescence. Fluorescence is the property of absorbing light rays of one particular wavelength and emitting rays with a different wavelength. Fluorescent dyes show up brightly under UV light as they convert into visible light. Coons et al. 1940 showed that labeled dyes can be conjugated to antibodies, and these labeled antibodies can be used to detect antigens. Dyes that are commonly used include fluorescein, phycoerythrin. So this is how direct and indirect immunofluorescence work. Cells with membrane antigens, primary antibody are shown here, and this is labeled with the fluorescent material. So direct method with fluorochrome labeled antibody to antigen. This is the how secondary anti-isotope antibody function. Indirect method with fluorochrome labeled anti-isotope type antibody. Here is the antigen, here is the primary antibody, here is the secondary antibody, and the fluorochrome is attached with the secondary antibody. So the primary antibody is specific to the antigen, and secondary antibodies are specific to the primary antibody. So indirect method with fluorochrome labeled anti-isotope antibody. Here is indirect method with fluorochrome labeled protein A. This is the antigen, the antibody attached. Protein A is attached to the primary antibody, and it is attached itself with the fluorochrome. So this is an example of indirect method. Application of antibodies, antigen antibody. There are multiple applications. You can have a look at this, and then finally the conclusion. So you can read this. If you have any question, feel free to contact me. I'll be happy to answer. Thanks for watching.